So we've started asking the question, what are those crazy little things called genes? And where we finished was with the work of a guy named Frederick Griffiths, who was studying bacteria, the bacteria that cause bacterial pneumonia. And he had made the discovery that you could mix dead bacteria of a type that normally made you sick, but was dead, and live bacteria of a harmless type, and you would get live harmful bacteria. And the only explanation was that there was some stuff, some substance that had gone from the dead bacteria to the living ones that transformed them from a harmless type to a harmful type. And he didn't know what this thing was. He called it the transforming principle. And then the work of a guy named Oswald Avery led to the identification of exactly what that transforming principle was. So did some work by a couple of scientists named Hershey and Chase, who did some beautiful experiments that I can't really get into right now because I haven't hit all of the background material yet. But Avery wasn't the only person who worked this out. The transforming principle turned out to be this stuff that was rich in nitrogen and phosphorus. That's the N and the P in schnapps, if you remember that from the very beginning of the course. And this stuff had already been discovered back in 1869 by a Swiss guy named Miescher. Prokaryotes and eukaryotes have it. In eukaryotes, it's restricted to the nucleus. In prokaryotes, there is no nucleus, and it's kind of just located in the middle of the cell. But because it's found in nuclei and it's a very weak acid, they called it nucleic acid, sensibly enough. This test tube actually shows some nucleic acid that's been extracted from probably strawberries. Down in the bottom of that test tube, there's soap. Up at the top, there's alcohol, and the light pink cotton candy stuff floating in the middle with air bubbles trapped in it. That is actually the nucleic acid. All this stuff right there. That's where the nucleic acid is floating. And it turns out that chromosomes are made up of proteins and nucleic acids. We will talk about proteins later. You've probably heard of them, if nowhere else, at least in the context of diet. You know, you've got to get enough protein and all that. And one of the things that chromosomes were made of turned out to be protein, but Avery was able to show that it's not protein that is this transforming principle, it's nucleic acids. All right, what are nucleic acids? All right, you probably have all had high school chemistry, and I'm assuming you know this already, but matter, you know, all things that have mass and take up space, solids, liquids, gases, etc., are made up of tiny particles called atoms. The exception is this really weird stuff called the Bose-Einstein condensate that we're not going to worry about. All of the matter that living things are made of that we experience under Earth conditions is made up of atoms. Can be solids, liquids, or gases. There's 88 naturally occurring types of atom, as well as about 30 odd that can only be made in laboratories. In other words, there are 88 natural chemical elements. And the chemical elements include the ones you should already know: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, schnapps. There are other elements that you need for life, like iron and zinc and things like that, calcium for your bones, and on and on. We've already talked about schnapps. Okay, 88 natural chemical elements, and they might I forget how far up they've gone. They're up to about 30 more that have only been found in, that it can only be made artificially. They don't naturally exist. Atoms bond with each other to form molecules. And if you have two or more atoms bonded together, we call that substance a chemical compound. For our purposes, we don't need to worry about how they bond together. If you want to know what makes atoms stick together, we've got a perfectly good chemistry department. For most of what we're going to do here, we can pretend that atoms are little ping pong balls stuck together with popsicle sticks. That's not really what's going on, but for what we need to do, that'll work. We're not going to need to worry too much about exactly what is going on in a chemical bond. That's one molecule of a chemical compound, and it happens to be called styrene. We've represented the carbon atoms by the letters C and atoms of hydrogen by the letters H. And every atom that's bonded to another atom, that bond is represented by a black line. And some of the carbons can form double bonds with each other. Carbon can do that. It's one of the things that makes carbon so incredibly flexible, so able to form such a huge number of different compounds. So we've got 6 carbons here in a ring. There's your ring of carbons. Some of them are single and some of them are double. There's some hydrogens sticking off right there. And then there's another pair of carbons up there with some hydrogens decorating them. And that compound is called styrene. Who cares? What I can do is take several styrene molecules and make them all join together in a chain. I can make bonds form between this carbon and that carbon. And I can make a bond form between this carbon and that carbon. And I could take this carbon and bond it to another carbon, and this one could bond to another. And we'd end up with a long chain of one styrene after another stuck together like cars in a freight train. And if I do that, it looks like this, and the resulting molecule is called polystyrene. And I picked polystyrene because if you make polystyrene and you blow air bubbles into it, what you get is styrofoam. Polystyrene is one of many types of plastic, and plastics are built like this. I could take a whole bunch of molecules of something called vinyl chloride, string them together, and I'd have something called polyvinyl chloride, otherwise known as PVC, which you've probably seen PVC. It's used to make pipes and things like that. Plastics are all made like this. Many biological molecules are made like this, and any large molecule that's made up of one or a few types of subunits strung together in a big chain is called a polymer. And each one of the subunits is a monomer. So this right here, this styrene that I've just circled would be one monomer. There's another monomer, that group of atoms. And this would be a third monomer, and there'd be lots of others, all of them strung together to make a polymer. So there you have several monomers. 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 Sorry, I don't know what came over me there. Right. You could think of a polymer molecule as being like a train. Think about this the next time you get stuck in traffic waiting for the Union Pacific train to pass by. There's a fairly limited number of cars on a freight train. You'd have box cars, you have those piggyback freighters, you know, that carry those big boxes that can be loaded directly onto an 18-wheeler. You might have tank cars. You might have flatbed cars. Yeah, I guess I caught the Muppet syndrome. At least I didn't catch George Jones disease. Sorry, I didn't catch Tom Jones disease. I don't know if any of you have ever had Tom Jones disease, but it's not unusual. Anyway, so there's a fairly limited number of types of car, but you can string them together in as many as 100 or more. I'm not sure what's the maximum size of a freight train, but you can put them together in a huge number of possible combinations to carry anything that needs carrying. You know, you can carry huge loads of many different kinds with the right combination of box cars and flat cars and piggyback freighters and, you know, tank cars and whatever it is that you've got. So you can think of polymer molecules as being like trains. They're very important in organisms. This is a molecule of cellulose, and you might remember that cellulose is the stuff that plant cell walls are made out of. By extension, it's what paper and wood and cotton are made out of. They're ultimately made of cellulose fibers. So you're probably, you know, if you're wearing a cotton shirt, writing on a piece of paper at a wooden desk, you're surrounded by cellulose. And what cellulose is a polymer of simple sugars. Each one of these hexagons right here, that's a simple sugar molecule. Now, we're actually leaving these out. Carbon is so common that we often leave it out, but there's a carbon atom at the corner of each one of these hexagons. And there's actually another one right there. So we've got 6 carbons and an oxygen making up this ring. And then there's actually some hydrogens that are stuck onto the carbon. These can often be left out for clarity. But that's what each one of these hexagons is. It's a molecule of a simple sugar of a type called glucose. And, you know, you might well have heard of glucose at some point. And if you take a bunch of glucose molecules and you stick them together, you get a really long fiber, a really long chain-shaped molecule. That's what cellulose is. If you stick those sugars together to make a slightly different type of polymer, you get starch. If you stick them together to make a type of polymer that you store in your liver, you get something called glycogen, which is one of the ways that your body stores energy. If you're an athlete, you might have heard of glycogen storage. Right. Chitin, the stuff that makes up fungus cell walls, is also a polymer of sugars. Proteins are polymers of simpler molecules called amino acids. Again, if you follow nutrition, you might have heard about amino acids. Those are what your cells have to string together to make proteins. And we'll be talking lots more about that later. Okay, nobody got my Tom Jones joke. But we'll be talking more about those. Nucleic acids also turned out to be a polymer. And in 1919, a guy whose name I'm not going to hold you responsible for, called Phoebus Levine, described the structure of nucleic acid and worked out that the monomers of nucleic acids, right, the simpler molecules that are strung together to make nucleic acids, were molecules called nucleotides. And this is what a nucleotide looks like. So you know, everywhere there is a corner or an angle, there's a carbon. And again, this is because if you know a bit of organic chemistry, it's so easy to predict exactly where the carbons will go that we can often leave them out. There's also some hydrogens in here that are getting left out. And, you know, again, there's a hydrogen there. And again, if you know a bit of chemistry, you find that it's often quite possible to leave hydrogens off for purposes of clarity. You can figure out where, you can always figure out where they go if they need to. But that's a nucleotide. And it turns out that every nucleotide is made of 3 parts. Just like every train car is made of parts, every nucleotide consists of 3 parts. There's a cluster of atoms over here that's made up of 1 atom of the element phosphorus and a bunch of oxygens and a few hydrogens. That's called the phosphate group. There's this ring right here that's made up of carbons, oxygens, and a few hydrogens. And that's a simple sugar, a little different from the sugar that's used to make starches and cellulose. And then stuck to the sugar, there's an either single or, in this case, a double ring-shaped molecule that contains a lot of nitrogen atoms. And we call that the base. So the phosphate group is over to the left, and I've shaded it in yellowish. The sugar is shaded here in green, and the base right here is shown in blue. So every nucleotide consists of a phosphate group, a sugar, and a base. Now, in the most important nucleic acid that we'll be discussing for now, there would normally, well, not normally, but there would often be 2 oxygen atoms sticking off it, one over here and one over here. And if we had 2 oxygen atoms, we would call this particular simple sugar ribose. Oops, sorry, there. However, in this particular nucleic acid, this is not ribose because we don't have an extra oxygen right there. There's just a hydrogen stuck there. There's the oxygen's missing. And so this particular sugar is called not ribose, but deoxyribose, meaning ribose, but an oxygen got taken away. This is deoxyribose. If you take a bunch of these nucleotides that contain the sugar deoxyribose and you string them together in a big chain, you get deoxyribonucleic acid. Or if that's too much of a mouthful, we can call this stuff DNA. So 1919, Phoebus Levine works out that the most common nucleic acid in cells is made up of these nucleotides. And because the sugar is missing an oxygen, we call it deoxyribonucleic acid. You can say DNA, of course, but that's what DNA actually means. DNA is what turns out to be the transforming principle. Those harmless bacteria were turning into deadly bacteria because it was DNA from the dead bacteria that were transforming them. DNA is the active ingredient in your chromosomes. And one thing that was soon found out is that DNA is not made of only one type of nucleotide, it's made up of 4. There's 4 different nucleotides, and they've all got the same arrangement of a sugar and a phosphate group. They've all got this, a sugar stuck to a phosphate. What makes them different from each other is that they've got 4 different possible bases. We usually can get away with abbreviating the bases with single letters. But one of the possible bases right here at the top left is a double ring of carbons and nitrogens and a few hydrogens, and we call that base adenine. Some nucleotides have a double ring of carbons that's got an extra oxygen stuck to it. That base happens to be called guanine, which we can just abbreviate G, as we abbreviate adenine A. The third possible base is a single ring of carbons and nitrogens with a couple of oxygens decorating it. That's called thymine, or we just abbreviate it T. And the fourth is another single ring that we abbreviate cytosine, or C for short. So when you're studying nucleotides, the scientists have to remember that the ribose is, or the deoxyribose is the same in all of them, and the phosphate is the same in all of them. So if you want to study what nucleotides do, you have to be all about the base, out the base. No sugar, I'm all about the base, No phosphate, I'm all about the base, out the base. Adenine and guanine and, right, sorry. Yeah, all about the base. Or does your generation remember that song? Probably wishes they didn't. Right. 1940s, guy by the name of Chargaff discovers some facts about DNA when he starts extracting DNA from different organisms and breaking it down. Turns out, first of all, that DNA from different species is different, which, all right, you might expect. Specifically, the percentage combination of nucleotides is different in different species. Your DNA happens to contain about 29% adenine. In corn, it's about 27. In octopus, it's about 33. You certainly don't need to remember those numbers, but let's just say that different organisms have slightly different compositions of DNA. All living things have some proportion of all four bases. There's no living things that don't use any one of the four, but exactly how much is there depends. So it's probably not surprising, but different species have different DNA, but there's rules to it. The percentage of adenine always turns out to be equal to the percentage of thymine, and the percentage of guanine always equals the percentage of cytosine. And simple algebra shows you can work out that the percentage of adenine plus guanine is always equal to the percentage of thymine plus cytosine. And we call those Chargaff's rules. Let me show you. Okay, I can't figure out how to type on these things, so I'm going to have to draw it. So let's say we have some human DNA, and the amount of adenine is equal to 29%. We know that the amount of thymine also has to be 29%, because the amount of adenine equals the amount of thymine. Okay, and we also know that the sum of all four nucleotides must add up to 100%. So if adenine is 29% and thymine is 29%, the percentage of adenine plus thymine is 58%. That means the percentage of guanine plus cytosine is equal to 42%, right? That's 100% minus 58%, right? Because A plus T plus G plus C has to be 100%. So if A plus T is 58%, G plus C is 42%, And since the amount of guanine is always equal to the amount of cytosine, the amount of guanine has to be 21%, because that's half of 42%. And the amount of cytosine also has to be 21%, because it's equal to the amount of guanine. So if you know only one of the percentage of only one of the 4 nucleotides, you can easily figure out the percentage of the other 3 using Chargaff's rules. Hint, you will see questions like that on the exam, where I tell you that such and such an organism has DNA that is 18% A, and you have to tell me the percentages of T, G, and C. You'll see questions like that again, and they're easy to solve if you just keep Chargaff's rules in mind. Chargaff at the time didn't know why DNA follows those rules, but we're about to find out. Picture this: Cambridge University, 1952. British scientist by the name of Francis Crick, who is pictured here on the left, and a visiting American named James Watson are working on DNA at Cambridge University in the lab of a senior scientist named Maurice Wilkins. And there they are. One of their colleagues in the lab is a woman named Ros Franklin. And Rosalind Franklin was one of the world's experts in a technique that had been developed at Cambridge and is still used, and it's called X-ray crystallography. What the heck is that? Okay, this had been first worked out, I want to say like 1909, something like that. We've known how to do this for over 100 years. And you can take a chemical and you make a crystal out of it. You could do this with salt. Maybe you've done it before, or sugar. If you take sugar syrup or a very salty salt water and just let it evaporate, you might have seen how the salt forms crystals or sugar forms crystals. If you've ever made rock candy at home, you might have seen how that happens. But you can take a lot of different chemical compounds and make them form crystals. And once you've done that, you shoot a focused beam of X-rays at one of these crystals. Now, when I was a kid, and we went to my grandmother's house for Christmas or for the summer, in her dining room, she had a crystal chandelier. And I loved looking at that thing because when the sunlight would pass through the window, a beam of sun would hit one of the crystals, and the crystal, of course, would sparkle and shine, and there'd be spots of light on the opposite wall. She also had, if you've ever seen sun catchers, they're just, pretty pieces of glass that you can hang up. And if you shine a beam of light through them, you get, little sparkles of light on the opposite wall. So if you've ever seen a crystal chandelier or just a glass sun catcher, you've seen how these work. Crystals do the same thing when you shoot X-rays at them. That crystal breaks up the beam. It's called diffraction into many beams. And your eyes cannot see X-rays, but photographic film can't. So you hit the crystal with an X-ray beam and you'll get lots of beams that strike a sheet of photographic film. And you get something that looks like this. Every one of these black dots in that picture is where an X-ray beam hit the film. And it takes a lot of rather gnarly mathematics. In this day and age, of course, computers do a lot of the heavy lifting. Calculating this by hand would be an absolute beast. But if you measure the positions of the dots on the film and the angles between them, it's actually possible to work out the position of the atoms in the crystal. That tells you the shape of the molecule that you're working with. That's a way that we can rattle our black box. You can't directly see molecules, and you certainly cannot directly see atoms. But for over 100 years, it's been possible to work out how big atoms are, what angles their bonds form at, and how they fit together in molecules using X-ray crystallography. It's hard, and Roz Franklin was one of the best in the world at doing it. So she could take something like this and tell you where the atoms are located in the molecule, whatever molecule this was. Well, Maurice Wilkins's lab was interested in DNA, and I want to introduce you to a concept we're going to be hitting for the next couple of weeks. To understand how things work, you have to understand their shape. You know, think about tools for a minute. The shape of, let's say, the head of a hammer determines what you can do with it, whether you use it for driving upholstery tacks or driving 10-penny nails or hammering hot iron on an anvil or any one of a number of other tasks that we make hammers do. The shape of kitchen knives determines whether you'd use them for cleaving meat or chopping vegetables or filleting fish. The shape of a screwdriver depends on, shows you whether you can use it for slot head screws or hexagonal head screws or Phillips screws. The shape of your tools determines what they can do. It is exactly the same thing with molecules. The shape of a molecule tells you what that molecule does. And so the clue here, the big black box that we have not yet opened, is what is the shape of the DNA molecule? We know it's a polymer. We know what nucleotides are. We know that when the DNA forms, the nucleotides obey some kind of rule, Chargaff's rules. But how does this all fit together? Franklin was able to make a DNA crystal, blast it with X-rays, and on the right, that's what she got. That's the photo. That's the photographic film that she got. And it may not look like much to you, and I can't make heads or tails of it. Franklin was able to work out that the DNA molecule has to be coiled. It's not just a straight string of nucleotides like beads on a necklace. The entire molecule has some kind of coil shape. Or as we say, it was a helix. A helix is just a fancy word for anything that's coiled. A slinky is a helix. A corkscrew is a helix. A bed spring is a helix. A spiral staircase is a helix. They're all helices. And Franklin was able to show that DNA was shaped like a helix, and she was able to work out how wide the molecule had to be and how tight the coils were, how close the coils were to each other. So she was able to work out the basic parameters of the shape. Watson and Crick got hold of Franklin's data, be it said they did this without her permission or knowledge. And so they knew that DNA was a helix. Franklin's data also showed how wide and how tight the helix was, and she was even able to show that the nucleotide phosphate groups had to be on the outside. They knew how big nucleotides were. They knew what shapes they had. They knew about Chargaff's rules because Chargaff had paid a personal visit to their lab. And so Watson and Crick started trying to build models that would fit all of these criteria. You say, we know what DNA is made of. We don't know how the pieces are put together, but once we put the pieces together, they have to fit Ross Franklin's data. So we can't just stick them together any which way. We've got to find a way to put nucleotides together using the basic laws of chemistry and Chargaff's rules and Franklin's data on the size and shape of the molecule. And they published a paper in April 1953 that described their model for the shape of DNA. Here they are. There's Watson on the left and Crick on the right, pointing to this model that they've made out of sticks. And it turns out DNA is not a single polymer. It is two polymers, two chains of nucleotides wrapping around each other, forming a structure that's not just a single helix, it's a double helix. So there they are pointing at their model of the double helix, and it may not be too clear what that is. So let's update this a little bit. On the left, that is a picture of DNA that shows just the chemical bonds. This is called a stick model. On the right, that's a model of DNA as a space-filling model where we're showing the atoms here as looking like ping pong balls. These are simply two different ways of showing exactly the same shape. And in both of these cases, the sugars and the phosphates are shown in white and pink, while the bases are red and yellow. So here, let me go to a highlighter. So this right here is one chain of sugars and phosphates. And stuck to the sugars and phosphates, we have bases. That's one strand of DNA. This right here, maybe I'll use a different color for that. Hang on. Yeah, this right here that I'm highlighting in green is the other strand of DNA right there. So we have two strands, and the backbone of each strand is the sugars and the phosphates, and then the bases are in the center of the molecule, and it's bonds between the bases that are holding the two strands together. All right, so that's what DNA turns out it looks like. If we could take a molecule of DNA and untwist it, it would look like a rope ladder. If you think about a rope ladder, the sides of the ladder, right, the parts that you're, the sides that are, you'd probably put your hands on, are the sugars and the phosphates, making this chain that goes, each phosphate binds with a sugar, which binds with a phosphate, binds with a sugar. So that's what's holding each strand together. I just highlighted that one in green. And let's do that one in yellow. There's that chain of sugars and phosphates. And then what makes the rungs of the ladder are bonds between the bases. So let's highlight those in red. Why not? So here's a bond between a guanine and a cytosine. And there's another. And there's another. And there's another. And so on. So we get this ladder-like structure to it, with the added wrinkle that the entire ladder is, actually, I didn't want to make that stroke there, but the entire ladder is twisted. There. So there we go. So the rungs are made by bonds between bases on separate strands. And then what holds each strand together is these backbones made up of the sugars and the phosphates. So each one of these right there, that's a nucleotide. And that's the next nucleotide. And that's the next nucleotide. And that's the next nucleotide. And of course, these are also nucleotides, nucleotides, nucleotides. All right, and then when the ladder twists into a spiral, we get the double helix, a short segment of which looks like this. In fact, using techniques called atomic force microscopy, it is now possible to confirm Watson and Crick's model and look at actual DNA molecules. C is maybe putting it a little bit strongly because we're not using light to take these pictures. You can't see molecules with light, but using a technique called atomic force microscopy, up here at A, this is a computer picture of an actual piece of DNA. B right here, this is the same picture that has been straightened digitally compared with that model right there. And you can see that that digitally straightened piece of DNA has a definite spiral shape to it. Another technique called scanning tunneling microscopy gives you even more magnification. And yeah, it shows you can actually see that double helix with the bases right there, right? Right there, there's one strand, there's the other strand kind of wrapped around it, and then right here you can see those bases. Watson and Crick basically got the structure right. Get it? Basically got the structure of DNA right. Get it? Because it's basic with bases, DNA bases. It's yes, so basic. It should be wearing UGG boots. Right. Okay. Thank you for the thank you for the pity laugh there. Right. Okay. Brief stepping outside of science into social issues that are raised. Watson Crick and Maurice Wilkins, who was their boss, shared the Nobel Prize in 1962, the Nobel Prize in medicine, for their discovery. And it turns out to be extremely important because, as I was saying earlier, the shape of things determines what you can do with them. And this double helix shape of DNA turns out to be absolutely critical for understanding how heredity works. This is what is inside one of that big black boxes. This is ultimately going to explain why Mendel got what he did with purple and white peas. Right? This has been a lot of information I have been socking you guys with over the past few weeks, but the pieces fit together. Think of this as doing a great big ass jigsaw puzzle, and it's all going to come together once we get a little bit more information about how DNA operates. I'll point out that Roz Franklin provided critical data. She didn't share the prize. It's sometimes it is said that she was robbed. In this case, probably not, but the Nobel Prize is not given posthumously. You can't give it to anyone after they're dead, and Roz Franklin died young, unfortunately. She died in 1958. So technically, Roz Franklin could not have actually shared the prize. That being said, her importance to the story has not always been recognized. Watson wrote a book in 1968 called The Double Helix. It was his personal account of making that discovery, and it's mostly considered a really good book because it gives you a real feel for what it is like to be on the cutting edge. You know, these days we're kind of blasé about it. You hear about DNA testing all the time. But in 1952, nobody really knew what this was. And, Watson and Crick were making a discovery that they knew was going to be extremely important. And the book really does give you the feel of what it was like. Watson, unfortunately, he's still alive, and he's still pretty much a bro. And in his book, he downplayed what Roz Franklin had done and mostly complains about the fact that she didn't dress nice. Watson is, I know some of you have, waited tables or worked retail. And those of you who have worked jobs like this and who are female, you ever had a customer who tells you that, you'd be prettier if you smiled more? That's Watson, okay? Watson is that really annoying guy who never tips. As he wrote in his book, The Double Helix, Franklin was not unattractive and might have been quite stunning had she taken even a mild interest in clothes. Like, who cares? She's a scientist, not a fashion model. There was never lipstick to contrast with her straight black hair, while at the age of 31, her dresses showed all the imagination of English blue stocking adolescence, which is another way of saying, she's not dressing flashy. Here's the thing. First of all, that's not even true. Roz Franklin never married, but she did have a lot of friends. Outside work hours, she was a lot of fun. She did love dressing up. She loved going to the theater, enjoyed plays and concerts and things like that. However, at work, she was all business. She did not come there to have fun. She dressed very conservatively. And did I mention that James Watson was really kind of a bro? She was, not mean, but serious about the work she was doing. She didn't goof around, and she didn't, as they say, she didn't suffer fools gladly. Right? She was, very professional. And it is a problem that women face in, do I mean a pervert? No, there's plenty of things you can call Watson that aren't very nice. As far as I know, he's not a pervert. As far as I know, he was pretty much just a jerk. That's not perversion. That's just being a jerk. And it's a problem women still face in the workplace because, many women report that, if you try to be nice and kind and smile a lot, people will walk all over you. And if you try to be, professional and, strictly business, no frills, get the job done, they think you're a bitch. The kind of assertive, take charge behavior that is praised in a male business executive is often considered, nasty in a female executive. There's that double standard, and it's still there. I would like to think that science has gotten better about it since then, although it's certainly not perfect. But Roz Franklin was kind of a victim here of that double standard. Be it said, she did deserve a large share of the credit. And Watson later did acknowledge her accomplishments. There's what Watson looks like today, not quite as much of a bro. He's holding a little double helix model, by the way. And as he wrote when he revised the double helix, we both came to appreciate greatly her personal honesty and generosity, realizing years too late the struggles that the intelligent woman faces to be accepted by a scientific world which often regards women as mere diversions from serious thinking. So he did eventually come around and admit that she was a good scientist who provided critical data for the discovery. If we could just get Watson to stop saying racist shit, it would be better. He's actually gotten himself into quite a bit of trouble recently for claiming that there's a genetic difference between Black and white folk on IQ tests. Short answer, no, there isn't. There is a lot of evidence that that has everything to do with the environment and very little that it has anything to do with racial genetics. So it's a shame about that. And remember, when I tell you the names of these people, I'm not expecting you to hold them up as heroes. They're important in the development of scientific ideas, but they're not necessarily role models. Yes, F that guy. Scientists are humans, and some scientists are pretty rotten humans. Some scientists are very decent humans, and most of us are somewhere in between. You know, that doesn't take away from the fact that Watson helped to make an extremely important discovery. But the fact that he made that extremely important discovery doesn't take away from the fact that he is pretty much a flaming sexist and racist. It happens, right? The guy who led the development of the transistor was, that made modern electronics possible, was a far worse racist than Watson is. So, people are people. Okay, so Watson the racist and Crick that rhymes with prick and Ross Franklin scoring a major assist worked out that DNA is shaped like this double helix. Who cares? Again, to understand how molecules work, you have to understand their shape. Understanding the shape of DNA is the key to understanding what it does and how it does it. Just like understanding the shape of a screwdriver head is the key to understanding what that screwdriver can do, or what you can do with the screwdriver anyway. And the same is true for understanding, understand the shape of a fish filleting knife or a blacksmith's hammer, or I don't know, any tool in Lowe's hardware. Understand the shape and you can understand what you do with it. So DNA, again, is a polymer, or two polymers wrapped around each other, of these subunits called nucleotides. So there's that picture again as a reminder. Every nucleotide has a phosphate group shaded here in orange, a sugar, in this case it's deoxyribose, shown here in green, and then one of four possible bases. What are the four possible bases? Fender Precision, Fender Jazz, Fretless, and Ibanez. Actually, I'm kidding. Not that kind of base. What holds the molecule together is this. Bases can form easily broken chemical bonds between each other that happen to be called hydrogen bonds. For our purposes here, we don't really have to worry about how hydrogen bonds work. If you want the full story, we've got a perfectly good chemistry department over in Mannion Hall. You can go take classes over there. For what we need to do here, you can think of hydrogen bonds as being like Velcro, right? If you want to attach two pieces of cloth together permanently, you'd sew them. And it's not very easy to, you can, but it's not that easy to take good sewing apart and separate two pieces of cloth. That's meant to be pretty much permanent. But you could take strips of Velcro and attach two pieces of cloth together and easily, separate the pieces of Velcro and easily put them back together anytime you wanted to. Zippers, it's the same thing. You can attach, the cloth on either side of your zipper, and your fly can be easily held together with the zipper and then easily unzipped when you need to unzip your pants. And you can think of DNA as being like a twisted zipper. The sides of the DNA molecule are not that easy to break down, just like it's not that easy to cut across a zipper. But the two halves of a zipper are easy to zip and unzip. That's the point of a zipper in the first place. And in the same way, those hydrogen bonds that are holding the bases of one strand next to the bases on the other strand, those are easy to break and easy to reform. Now, because of the way the bases are shaped, if there's an A on one strand, an adenine, it forms hydrogen bonds with a thymine on the opposite strand, and it only forms hydrogen bonds with a thymine on the opposite strand. And vice versa, a thymine on one strand can only bond with an adenine. A guanine on one strand can only bond with a cytosine on the other strand. And we call that base pair complementarity. So A only forms a hydrogen bond with a T, and a G only forms a hydrogen bond with a C. Now we've got an explanation for Chargaff's rules, right? For every A on one strand, there has to be a T, and for every G on one strand, there has to be a C. So this DNA consists of, what have we got? We've got 1, 2, 3, 4, 5, 6 Gs. And for every G, there has to be a C. So we've got 6 Gs and 6 Cs. The amount of Cs and Gs are equal. And then we have 2 As, and each A forms a bond only with a T. So for every A, there's a T, right? The amount of, we have 2 As and 2 Ts, and we have 6 Gs and 6 Cs. That's why Chargaff's rules work. If we somehow, took out one of these T's and replaced it with a G, we could do that, but these hydrogen bonds wouldn't work properly, and we'd have the equivalent of a zipper with a broken tooth, right? You ever had to deal with one of those where some of the teeth on the zipper don't perfectly fit? We'd get the same thing with this DNA. The DNA would not have the right shape. The DNA would ultimately have to be repaired, and maybe I can talk about how that works a little bit later. So that base pair complementarity is the reason why Chargaff's rules work. And it also is what happens in that mysterious S phase of mitosis. If you remember that, remember that every chromosome is made of DNA and proteins. In fact, every chromosome is one great big long piece of DNA. When a cell begins to divide and the cell goes into prophase, the DNA starts coiling, and it coils so tight that it becomes a visible lump. When the DNA isn't coiled, you can't see the individual chromosomes for the same reason that you can't count individual noodles if you're served a giant bowl of spaghetti, right? The noodles are not neat and compact, they are tangled every which way. But when prophase happens, the long pieces of DNA that make up the chromosomes coil in on each other, and they become so compact that it's now possible to see them. That's what is going on in mitosis, we now know. If we could take one of your cells and extract all 46 chromosomes and unwind them all and put the DNA end to end, each one of your cells would contain about 6 feet of DNA, about 2 meters, for those that like the metric system. So now we've got what's happening in that mysterious S phase. That's the point when DNA gets replicated. When DNA replicates, those hydrogen bonds that are holding the strands together get unzipped. And the 2 unzipped strands move apart. So there. Now think about this. New nucleotides come in and they get assembled. We'll talk later as to what does the work of assembling them. but new nucleotides come in and they get assembled into new strands that are complementary to these two old strands. So here's a G. Many nucleotides might come in and try to fit this G, but the only one that can make that hydrogen bond is a C. And then the only one here that can make a hydrogen bond with the G is a C. And there's a C. And the only one that can fit with that A right there is a T nucleotide. And the only one that fits that T is an A. And then the only nucleotide that will fit with that C is a G. And so only G's will fit those C's right here. And then when all of these get welded together, we have a new strand right there that matches the old strand over here. And the same thing happens over in this direction. Every G pairs up with a C, every A with a T, every T with an A, and every G every C pairs up with a G. And then when those fit together, we have a new strand that's identical to the old strand over on the left. In fact, that reminds me of the time I walked into one of my colleagues' labs. And while he's busy, I'm just standing around. And as I'm standing there in his lab, I hear this test tube say, "Hey, man, that shirt looks great on you." And this is weird. I've never heard a test tube talk to me before. And I think I'm going crazy. But there it is again. The test tube says, "Oh, man, you are looking really good today. Are you working out?" And then when my colleagues got a minute, I tell him, "Hey, man, did you know that test tube just talked to me?" And he said, "Yeah, I mixed guanine and cytosine together in that test tube, and they're complementary." Okay, right. I'm imagining you all laughing hysterically.
That's
All right.
hilarious.
Thank you, thank you, thank you. Thank you very much. Okay, all right. Just making sure, trying to keep everybody's interest up with the jokes. So here's computer animation. Each one of these new strands uses the old strand as a pattern. And the end result is we have two identical strands of DNA, right? They're identical. The order of bases on one of these strands is used as a pattern to assemble the complementary strand. No, Ian, it's nucleic acid, not lysergic acid. Okay, so every strand of a DNA molecule is used as a template, as a pattern to make the complementary strands. When DNA replicates, it unzips up here, hydrogen bonds break, and then each strand becomes the pattern for assembling a new strand. The actual work of assembly is done by a couple of enzymes called DNA polymerase and DNA ligase. We will worry later about what enzymes are. Don't worry about it just yet. You could think of them as little fairies that do the work. And again, every chromosome is a single long piece of DNA. This is an actual human chromosome that is partly unwound. When a cell is in interphase, the DNA is completely unwound, and you cannot make out individual molecules of the stuff. Again, because you're looking at the equivalent of a giant bowl of spaghetti with all of the noodles, intertwined with eeach other so tightly that you can't really tell where one noodle ends and the other begins, right? You can't count, you can't count individual noodles just by looking. But when prophase kicks in, what is going on during prophase is that DNA starts wrapping itself around a core of special proteins. And so you get a very tight coil of DNA wrapped around proteins. and then the coil coils on itself, and then that coiled coil coils on itself again. It would be like if somebody has really, really long hair, imagine you made a bunch of small braids and then braided the braids into several larger braids, and then made a braid out of the braids of braids into one, super braid ponytail. That's kind of what DNA is doing here. It coils, and then the coils coil, and the coils of coils coil, and then the coiled coils of coils coil, until you get this very compact structure. That's a chromosome pictured with a gizmo called a transmission electron microscope that has been completely uncoiled. The proteins is this stuff right here, and you can still see the shape of the original chromatids there and there. That's the protein core of a chromosome during mitosis. But all the DNA has become unspooled. And so all of this tangled spaghetti-like stuff, you can even see individual strands of it up there, have become released. So that's what a chromosome is. It's a giant piece of DNA, and DNA replication, it's what's going on during that S phase of the cell cycle. Each chromosome is one long piece of DNA, and it replicates in S phase to form two copies, two chromatids joined at the centromere right there. All right, and I've got 10:39, so I am going to stop recording.